Okay, so remember last part we were talking about the Plasmodial slime molds and their features and their characteristics. And I left off telling you I was going to show you guys a little story or a little situation that happened at my house. So quite a few years ago, I lived in a house where the garage had mulch right up to the foundation of the garage. So there was the mulch and then the concrete of the garage. And one day I went out there and I looked and I discovered this growing on my garage. I thought, well, that looks funny. It almost looks like that spray foam you would spray if you had a crack or something. It's called grate stuff. And you would spray it and it expands out. But then I started poking around and kind of looking at it. I'm like, oh my goodness, this is a plasmodial slime mold. You can see the little tendrils of the body here and the plasmodium. And what happened is this showed up. It was in early summer, towards middle of summer, so it's starting to get hot and dry. And what the slime mold was doing was starting to aggregate and come together and kind of solidify itself because it was getting hot and dry and it was getting stressed. So it was, like I said, this was attached to the side of the concrete on the garage foundation. And in the month of July, I would see it. And then late July, early August, it disappeared. I thought, well, that's weird. Maybe it just moved on or died or whatever. Thought nothing of it. So what happened is this thing got stressed, formed the sporangia, and then the body of the organism died, disappeared, came off the garage. But then come October, when it cooled down and we started getting some rain in the fall, it came back. Well, let me rephrase that. The next generation came back because it had shot out millions of spores into the mulch next to the foundation of the garage. And about 6 inches next to where this one was at, a new one had started to grow and develop. So it didn't really get far away, but in the plasmodial slime mold world, 6 inches is a pretty significant distance. So it had recognized environmental stress. I'm going to die. Time to reproduce. So when the slime molds go through their life cycle, they can run this haploid diploid life cycle where the thing you tend to see is the diploid portion of it, where it's got the full genetic material, it's building up, eating stuff, crawling all over things. And then it says, okay, time to reproduce, usually because of stress. And I'm going to form these little stalk structures called sporangium, little bulb on the top. That look like blueberries in the other one, whatever you want to call these things here. And then it releases spores. So spores are produced through the process of meiosis. Spores are all coming out as haploid structures, half the genetic material. Spores hit the ground in a new, better environment. They germinate, and then those little flagellated cells, you got one over here, you got one over here, they can get together and combine, fertilize to create the zygote, which is then going to be the diploid organism. So parallel in the animal world, you got egg and sperm. They combine, form the new offspring, and the life cycle continues. So, okay. So again, kind of a weird organism, but you never know where you're going to find it. If you guys have a pile of leaves that have been sitting on the ground for a long time, or a piece of chunk of wood, tree, a branch or something, lift it up. And I bet you might find this stuff sitting underneath it. Moist, terrestrial environments, decomposers, breaking down stuff, eating bacteria, yeast, microorganisms, things like that. All right, so an important part of a terrestrial ecosystem. All right, so this brings us to our third group within the amoebozoas, the cellular slime molds. All right, so weirdness here. They all have the pseudopodia with the actin and myosin, but they can be, these are the Power Rangers. I'm going to actually put that up here so you guys remember this. So if you want to sing the theme song, if you guys ever watched Power Rangers, and remember them when we talk about these guys. All right, so big things when we're looking at the cellular slime molds. They can all act as an individual, like amoebas. But what's really unique and actually really cool about these guys is that under stress, they will form a multicellular organism called a slug. So that's what you're looking at here is the slug forming, and then it progresses to the structure over here where it then has the ability to reproduce. All right, so I'll get into that. I'll show you some pictures in a second. So a couple other things we want to note and mention about cellular slime molds, because some of you guys may use these in the future depending upon what you do with your biology degree. Okay. Actually, let me. All right. So these guys are considered a model study organism for what's called differentiation. So cells go through a division process and divide, and at a certain point, they get their job. That's known as differentiation. Okay, so in humans, think about stem cells. Stem cells can do anything until they get their job when they've differentiated. Well, that's what we're using cellular slime molds to study is differentiation process. And if we can understand it in them, that helps us understand and use that knowledge for our stem cell research. So again, you guys may see this in your future depending upon what you do. What do these guys do in the environment? We would say they're consumers or omnivores. They eat soil nutrients, bacteria, I'm just going to say etc. So kind of whatever they can get a hold of, they're going to consume it. So they wrap around it. They're that amoeba-like structure with the pseudopodia, engulf it, and then consume it. But again, the really cool thing is during stress, they form that multi-organism, what we call the slug. And then the slug can differentiate into a thing called the saurocarp. The saurocarp's the reproductive structure. It's going to shoot out spores. And the goal is to get them as far away as possible, so hopefully those things germinate into new individuals and they have success. So let's look at the picture. The diagram is much easier to do it that way versus me trying to describe it to you guys. So, okay, so here is the process. Starts out as the basic structure here, the individual, and then during stress, it sends out a signal. And that's one thing that's really, really cool and not clear. So you have these simple blob things that can communicate. It's thought the communication is all through pheromones, chemical communications being released into the environment. Other individuals pick it up and recognize it and say, oh, okay, George said it's time for us to get together because the environment's going to, we need to start moving together, find each other, and then aggregate into this multicellular structure. So that's what they're doing here is aggregating. Once they get enough of them together, then they can actually form the saurocarp. This is the process of the saurocarp forming. Let me get the draw out. When they start going like this, to here, to here, to here, and then eventually to here. And the key is, notice it's getting taller and skinnier. And up top is where all the little spores are going to be housed. And the hope is, let's pop and release those spores, and hopefully they land as far away as possible, because where we're living right now, that environment is not good. The environmental conditions are not doing us any favors. This is just the saurocarp after the spores have been released over here. It's done. And now the environmental conditions may shift, and the original slime mold dies, but the spores are sitting out there. Spores are a great protective structure. They can put them into a period of dormancy and let them kind of hang out for a while until environmental conditions get better, and then it triggers their development out of the spore stage. So again, think about it. This is just amazing that this slime mold can communicate and do all this. No brain, no neural system, but it has all these abilities. So it's a pretty amazing feat or feature for what we would consider a very simple organism. All right, so let's do our review. Here's the review slide for us. So again, as I'm continuously going to ask you guys, what is the key feature to put something in supergroup Amoebozoa? Once you're in Amoebozoa, what brings you over here to amoeba? What are the features or unique characteristics? What makes something a plasmodial slime mold? Features or characteristics? And finally, what makes something a cellular slime mold? Features, characteristics that make each of these subcategories of Amoebozoa unique and different within the Amoebozoa supergroup. So it's the dichotomous key idea constantly when we're looking at supergroups and classification with the Protist. Keep that concept in mind, and that hopefully will make life so much easier for you here. Okay? All right, now our last supergroup. Let's get to it. The last supergroup, let's go with orange, is Opsitancanta. And no, I can't say it correctly. There's 100 ways to say it. But this is our last supergroup to take a look at. Now, we are not going to worry about fungi under this supergroup or animals under this supergroup. We will approach both of those independently as kingdoms. The group we're going to talk about under Opsicanta are the Choanoflagellata. So this is the group we want to look at. But what we want to figure out is what makes something go into this supergroup? Why are fungi, animals, and these things called choanoflagellates all grouped together? Well, the big key feature is that they have propulsion by a single flagella. That is the supergroup feature for the obstacants that I want you guys to remember here. Okay? So they move, they're mobile, but they're using a single flagella for that movement. Within this group, choanoflagellates are the ones we want to talk about here. Single-celled sounds so much better than unicellular. So, okay, single-celled choanoflagellates are all single-celled organisms. They all are individual. But what they can do is they can aggregate together to form a multicellular organism. So here's one with a little flagella, here's another with a flagella, another with a flagella, and keep doing this all the way around. So they aggregate together, kind of like that cellular slime mold we just got done talking about. All right, so these guys are the ancestors to sponges. Sponges are animals, though. They go under kingdom Animalia. So the choanoflagellates gave rise to some of the earliest, simplest animals, those being the sponges. So when we get to animals and we talk about sponges, I will ask you, who is the ancestor to the sponges? Come back to choanoflagellates. This is why I'm covering protists early in the semester, because plants, fungi, plants, and animals all evolved from protist ancestors. Okay, so that's it on the obstacons at this point. I said fungi and animals we'll tackle differently. I'm going to break it here, and then the last part of this protist lecture, I want to take a quick run through like the ecological aspect of the protists. There's some really, really important things to note about the ecological features and aspects of the protists.